Welcome everybody to the miRNAscope webinar from BioTechne. My name is Amrita Roy. I'm a field application scientist here at BioTechne, and I'll be your moderator for today's event. I would like to thank everyone for attending today. Here is our agenda for the webinar. First, an introduction to the miRNAscope technology and workflow, followed by the key applications that necessitate the use of miRNAscope assay and the key benefits of this assay for your research. Finally, we will hold a live discussion answering your questions at the end of this event. This webinar is supported by BioTechne. BioTechne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tokres Bioscience, Protein Simple, Advanced Cell Diagnostics, and Exosome Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments. You can find more information about BioTechne's innovative solutions at biotechne.com. Before moving into the talks, I have just a few housekeeping items to address. Throughout the webinar, we would invite you to submit any questions you have for our speakers or panel using the questions box in the GoToWebinar control panel. These questions will be asked directly following the presentations. Throughout the webinar, we will have polls and we would request you to participate in the polling. We will share the results after each polling. And now I would like to introduce our speakers for today, Dr. Miguel Mata and Ms. Helly Pimentel. Miguel is a scientist at Advanced Cell Diagnostics, leading the development of the miRNAscope assay. He earned his Ph.D. degree from the University of Texas Southwestern Medical Center, followed by a postdoctoral fellowship at Stanford. Miguel has over 15 years in research experience with a particular focus on assay development and molecular virology. Helly is a product manager at Advanced Cell Diagnostics, overseeing the miRNAscope product line. She earned her bachelor's degree in the field of biochemistry and molecular biology, followed by an MBA degree. Helly has an extensive biopharma industry experience for almost 12 years. And now I'll invite both the speakers to start the presentation.
Hello, everyone. Thank you for joining our webinar. My name is Helly Pimentel, and I'm a product manager at Advanced Cell Diagnostics, abbreviated as ACD. ACD is a division of BioTechne Corporation. My colleague Miguel Mata, a scientist from our R&D department, will be co-hosting with me today. For today's webinar, we're delighted to introduce you to ACD's newest product offering called the miRNAscope assay, specifically designed for the detection of small non-coding RNAs, such as microRNAs, and short therapeutic oligos, such as small interfering RNAs and antisense oligos. On the agenda for today, we'll first introduce you to ACD's product portfolio for the use of a wide range of applications. Then we'll switch gear and introduce you to the newest miRNAscope assay to detect smaller RNAs. After that, my colleague Miguel will present on the common applications that necessitate the use of the miRNAscope assay, along with some beautiful data to share with you. Additionally, we'll look at the assay controls and provide our staining scoring recommendations. Towards the end, we'll talk about the product offerings as well as in-house assay services that are available for your research. Finally, we'll summarize our presentation and address some of your questions. To begin, I would like to first talk about the importance of single-cell transcriptomics with spatial analysis. Previous attempts at detecting RNAs have relied heavily on traditional bulk genomic analysis tools, such as qPCR or microarrays. Although these methods deliver bulk expression levels, they do not provide detailed spatial information, nor gene expression at subcellular level. Similarly, single-cell sequencing methods could offer a single-cell resolution, but still lack the important spatial resolution that researchers need to understand gene localization. So that brings us to ACD technology, where our advanced in situ hybridization technology enables single-cell spatial analysis of any genes from any species, allowing analysis of where exactly every single molecule is located relative to every other molecule in the tissue sample. These spatial and single-cell information is important for understanding pathological relevance of transcriptomes. ACD's core technology is the RNAscope technology. RNAscope is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single-cell resolution. The RNAscope technology consists of 3 parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots for quantitative analysis. The assay allows for the spatial mapping of mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, point mutations, all the way to small RNAs, such as microRNAs and siRNAs, and cells and intact tissues, all which can be visualized with either fluorescent or chromogenic labels. In addition, the assay can be performed on a variety of sample types, including FFP tissue, fresh frozen or fixed frozen tissues, and culture cells. Next, focusing on the core RNAscope technology workflow. The RNAscope workflow is similar to immunohistochemistry, so researchers who are familiar with the tissue-based protein assays find it quite intuitive to adopt this workflow. First, we start with properly permeabilized cells or tissues with ACD's pretreatment reagents. Then we hybridize the unique probe that ACD designed to the target RNA of interest. Next, with addition of the proprietary amplification system in the form of detection reagents, which allows the visualization of a single RNA molecule in the form of dots and enables us to quantify the level of gene expression indirectly. The combination of probe design strategy and the amplification system allows for high signal-to-noise detection of the target RNA. Our products cater to each of these steps, providing ready-to-use reagents in most cases. To design a specific probe for your target of interest, ACD requests you to provide us either the accession number information or the exact sequence of the target. You could even indicate your region of interest for targeting with start or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the sequence composition of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNAscope conditions. Next, ACD can design probes for virtually any species and have done so in over 200 species, covering most applications that are highlighted on our website. For example, the majority of our probes are designed in human and mouse. On our website, you'll be able to find the applications area to demonstrate how these probes are used in the most common applications, including infectious diseases, cancer, neuroscience, and gene therapy. We've also added support for signaling pathways such as WNT or JAK-STAT and indicated the probes we have available within that pathway. In this slide, we're looking at the 3 unique assays that ACD offers to detect any RNA with spatial mapping at single-cell resolution. First, on the left, RNAscope is designed to detect any mRNAs or non-coding RNAs that are greater than 300 nucleotides in length. RNAscope probes are designed in about 20 ZZ probe pairs, and the assays are available in either chromogenic or fluorescence detection modalities for up to 12 targets simultaneously in a single slide. On the other hand, the BaseScope platform listed here in the middle uses probes that are designed in 1 to 3 ZZ probe pairs to specifically support the short target detection of splice variants, highly homologous sequences, point mutations, or short targets that are 50 to 300 nucleotides. The BaseScope assay can detect up to 2 targets with the chromogenic assay. Last but not least, ACD offers the newest miRNAscope technology to detect even the smaller RNAs and oligos, such as microRNAs, antisense oligos, small interfering RNAs, or other sequences in the range of 17 to 50 nucleotides. This new assay is currently available as a chromogenic singleplex assay. Now, this slide summarizes all the assay products that are currently available from ACD. We have chromogenic singleplex brown or red assays and duplex assays on both the manual and automated platforms. The multiplex fluorescent assays allow for the detection of up to 4 targets simultaneously in a single tissue section. The major difference between the two fluorescent assays is that the V2 assay uses fluorophores from Akoya Biosciences for signal visualization and provides a high signal-to-noise ratio, whereas the fluorescent multiplex assay is an all-in-one kit. The V2 fluorescent assay is also available on the Leica automation platform. For higher plexing, we have the RNAscope HighPlex assay, which enables the detection of up to 12 targets simultaneously in a single tissue section. Currently, the HighPlex assay is available in the manual format only. If your target of interest is shorter than 300 nucleotides and studying splice variants or point mutations, then the BaseScope assays will be the right choice for you. Currently, the BaseScope singleplex assay is available on all platforms, while the duplex assay is available on the manual platform. Finally, expanding our product portfolio, not only do we have assays to detect mRNAs, non-coding RNAs, short targets, splice variants, and point mutations, but we now have the miRNAscope assays for the detection of microRNAs, small interfering RNAs, antisense oligos, or other smaller RNAs, as low as 17 nucleotides in length. As of today, we have over 26,000 unique target probes in our catalog. Custom probes can also be designed against any targets from any species in as little as 2 weeks. For the remaining webinar, we'll bring our focus to ACD's newest miRNAscope assay. Overall, the miRNAscope assay is a robust in situ hybridization assay, enabling highly sensitive and specific detection of small non-coding RNAs, such as microRNAs, short oligos such as ASOs, and siRNAs in tissues with spatial and morphological context at single-cell resolution. In addition to these key applications that I just mentioned, the assay will also enable the detection of any smaller RNAs that are 17 to 50 nucleotides in length. Some popular targets include tRNAs and pVRNAs. Key research areas for these top targets include cancer research, infectious diseases, neuroscience, and therapeutic target validation. Additionally, this new assay leverages ACD's core amplification system and probe design capabilities to enable the detection of RNA molecules at single-cell resolution while improving high signal-to-noise detection of smaller RNAs with ease of data interpretation. To enable further insights into your research, the miRNAscope assay is also compatible with immunohistochemistry or immunofluorescence assays, allowing the detection of these small RNAs and proteins simultaneously on the same tissue slide. Next, what are the specifics of the miRNAscope probe design that makes the assay highly specific and sensitive? The miRNAscope probe design is a brand new design strategy for leveraging ACD's core RNAscope technology that the design algorithm screens the whole transcriptome to avoid any cross-hybridization. Due to the extremely short sequences of these targets, sometimes cross-hybridization is unavoidable. So in the cases of potential cross-hybridization, this information will be solely disclosed to our users for evaluation. However, I would like to mention that the majority of these cross-detection targets are endogenous transcripts, so we highly recommend our users to always include a negative control as the baseline to interpret their data. For microRNAs with cross detection, we recommend the use of a no negative control sample for the microRNA target. On the other hand, if your targets are ASOs or siRNAs, we always recommend in your studies to include a mock or untreated sample for your data interpretation. In addition to the new probe design strategy, we also improved the probe hybridization buffer to deliver optimal assay performance. Together with these conditions, the miRNAscope assay offers superior sensitivity and specificity without the need to further optimize the target probe hybridization conditions. For the miRNAscope assay, we also offer a universal scramble sequence probe as the negative control probe to evaluate specificity of the assay. Additionally, you could also request a specific scramble sequence to be designed against your smaller RNA. As I just mentioned, one of the key features of the miRNAscope assay is the probe design. The oligonucleotide target-specific probes are designed using an informatics algorithm that selects sequences to specifically bind to the target sequence and not cross-hybridize with any other sequences. The bottom of the probe design complements and hybridizes to the target transcripts, whereas the top of the probe is the base for the amplification structure, upon which a preamplifier can bind and the amplification tree can then be built. Once the preamplifiers are hybridized to the target probe, multiple amplifiers are then added to the preamplifiers. Each amplifier can then further bind multiple label probes, sequentially hybridizing to assemble a branching complex at each probe design probe binding site. Label probes contain a chromogenic enzyme that generates a visible signal after chromogenic reaction, in this case with fast red, that is detectable under a standard bright field microscope. This entire amplification system allows for highly sensitive and specific visualization of target RNAs as puntate dots. Next, we'll focus on the overview of the miRNAscope assay workflow. The new miRNAscope assay workflow is very similar to RNAscope singleplex workflow for those of you who are familiar with our technology. First, the cells or tissues that are bound to a slide is permeabilized using ACD's ready-to-use pretreatment reagents. The new assay supports a variety of sample types, including FFPE, fresh and fixed frozen, and culture cells. Then, the smaller RNA target probes are hybridized to the target RNA. Currently, the miRNAscope assay allows for a single RNA target detection. The probe hybridization step is universal across all targets, meaning further probe optimization is not required. After probe hybridization, the target probes will be hybridized with ACD's modified branch signal amplification tree, consisting of amplifiers and label probes to provide high sensitivity and specificity. After the colors have been developed, the small RNA molecule signal can be seen as red chromogenic deposits using bright field microscopy. I'd also like to mention that the fast red chromogen can also fluoresce similar to the ADO 550 dye under a fluorescence microscope. These slides can then also be scanned for further qualitative or quantitative analysis. The entire singleplex assay workflow can be done either manually or fully automated using the Leica systems. Switching gear, as I mentioned in a previous slide that the miRNAscope assay is compatible with multiple tissue types. On this slide, we're demonstrating robust assay performance in various species of formalin-fixed paraffin-embedded tissue samples. For example, demonstrated here, microRNA 124 3P is detected in mouse, rat, and human brain tissues with abundant signals detected. To demonstrate specificity, a known microRNA 124 negative tissue, such as the mouse liver, is also stained with the microRNA 124 probe. As you can see here, the negative tissue shows no background detection of the microRNA 124 target. Additionally, the miRNAscope assay demonstrates similar detection robustness with fresh frozen tissue samples. Demonstrated here in the left column, microRNA 124 is stained in the fresh frozen mouse brain tissue with abundant signals similar to what we just saw with the FFPE tissues. Again, the scramble probe is used as the negative control to demonstrate assay specificity, here showing no background was detected. On the right column is another example of fresh frozen tissue, but with microRNA 122 stained in the mouse liver, showing robust detection of the target and no background was detected with the scramble negative control. On the other hand, if you're working with culture adhering cells, the miRNAscope assay also performs extremely well as demonstrated in this slide with miRNA 205 stained in A431 skin cells. The assay successfully detected the abundant miRNA 205 signals in the skin cells, and again, the scramble probe was used as a negative control. Overall, the miRNAscope assay is a highly robust assay that can work with any of your sample types. So why choose ACD's miRNAscope assay? The miRNAscope assay offers many benefits for your research. First of all, the assay enables highly specific and sensitive detection of your smaller RNA targets at single-cell resolution while preserving spatial and morphological context. Additionally, the assay is compatible with multiple sample types, including FFP tissues, fixed and fresh frozen tissues, and cultured cells. Another benefit of the miRNAscope assay is that it does not require an RNA-free environment like other typical assays do. Furthermore, the assay reagents also offer in ready-to-use formats for ease of use. On top of that, ACD offers a one-on-one scientific consultation with our expert technical support scientists to ensure that you have a successful run of the assay. In addition, the miRNAscope assay is compatible with IHC staining, enabling co-detection of RNA and protein in a single tissue slide. Last but not least, the miRNAscope assay uses universal assay conditions where minimum optimizations are required for a successful detection of your target. Prior to commercialization of the product, we have had many successful customer stories using the miRNAscope assay through our in-house pharma assay services team. For example, one of our customers had used the miRNAscope assay to detect their siRNAs and provided us with an extremely valuable feedback, stating that the assay is incredibly specific and that the results are very easy to understand. The ACD Pharma Assay Services team had generated impeccable tissue staining for their siRNA therapeutic research and development. Another successful customer who had used our miRNAscope assay to detect their ASO says that they use ACD's assay to detect both the RNA and short oligos in serial tissue sections, which generated signals that are both evident and specific. And with that, I'll hand it over to my colleague, Miguel Mata, to provide you with detailed presentation on how the miRNAscope assay could help with your research applications.
Thank you, Helly. That was a great introduction on the miRNAscope platform. And as you mentioned, now we'll go into more detail related to the key applications of this new-ish assay that we recently launched a couple of weeks ago. So the key targets for this technology, and I will describe a few of them, a few examples of this, include the detection of this at the single-cell resolution of this small non-coding regulatory RNAs that fall between the 15 to 15 nucleotides in range. The short RNA nucleotides, oligonucleotides include antisense oligonucleotides or ASOs, small interfering RNAs, siRNAs, or small hairpin RNAs or shRNAs. And these are the synthetically designed oligonucleotides. And this particular oligonucleotides can target specific disease-related transcripts. And by doing so, they can transcriptionally downregulate these targets that reduce eventually the bioavailable levels of a particular protein of interest. And in the past few years, the small RNAs have actually gained a lot of interest as potential therapeutic agents in a number of diseases. Next is microRNAs, and these are also small non-coding RNAs, and they function in RNA silencing, more specifically at the post-transcriptional gene silencing level. Also, piRNAs, and these are also short RNA species that are slightly larger than microRNAs, between 26 to 34 nucleotides. And they have been associated with silencing transposable elements within the germline. And also in somatic tissues, they regulate mRNA expression at the level of the post-transcriptional stages. So these are the next the next application is ISH and in situ hybridization or immunofluorescence. Now, miRNAscope workflow is fully compatible with IHC or IF. Now, this is an extremely important application if there is a need to identify a particular cell type of interest, or even to determine the levels of a particular protein whose transcript was originally targeted by one of the aforementioned regulatory small RNAs. Now, using miRNAscope, you can specifically identify any of these subtypes of small RNAs, and then using IHC, you can identify a particular cell type of interest. Now, these are all the key research areas where miRNAscope has made an enormous impact. First is basic science. This is in terms of neuroscience, stem cell, and developmental biology research. Next is in terms of infectious diseases, in particular with a focus on viral infections and also immune cell regulation. Next, in terms of biopharma therapeutics, as I mentioned, siRNAs and ASOs have gained a lot of attention as novel therapeutic agents, and we'll discuss a few of them in the later parts of the webinar today. And also cancer and clinical research. Here we will discuss the role of microRNAs and these other regulatory RNAs in terms of immuno-oncology and tumor biology. So let's move to our first application, which is microRNAs and their role in different research areas. So some of the research areas include oncology, neuroscience, and immunology. So how is microRNA detection relevant to oncology? So microRNAs can actually alter the transcriptional landscape within single cells by targeting key players or even signaling pathways. And in this way, those pathways may have some function in malignant transformation. Some targeted microRNAs have actually been identified as oncomeres, or a microRNA that has the ability to promote malignant transformation. There's also, on the other hand, a different class of microRNAs that actually have tumor suppressor characteristics. In addition, numerous microRNAs have also been identified as potential biomarkers for a number of malignancies. What about neuroscience? How is microRNA detection relevant? As it turns out, there are numerous microRNAs that have been identified to be deregulated in different neurological disorders. And many of these studies have actually implicated microRNAs as potential biomarkers as well for neurodegenerative diseases. So it will be important to identify those key microRNAs with the spatial and the morphological context that miRNAscope provides. Also, in immunology, there's particular importance in terms of infectious diseases. It is known that microRNAs regulate immune activation of different pathways that are related to the immune system. So failing to turn these pathways on or off can actually have severe consequences. Next, we will talk about specific microRNAs in key areas. So the first area is oncology. So here, this slide is showing a number of microRNA targets that were, of course, detected using the miRNAscope assay and that have been implicated in some cancers. Here, we're showing two different cancers, head and neck cancer on the bottom and cervical cancer on the top. Now, these microRNAs have previously been implicated in cancer initiation or progression, and they do so by deregulating signaling networks, as I mentioned previously, that may lead to malignant transformation. Now, the first microRNA that we have here is miR205-5P, your most left. And this particular microRNA is interesting as one of the most popular microRNAs studied in oncology because it's known to promote tumor invasion, migration, angiogenesis, and it does so in part by activating the AKT signaling pathway. Next is miR21-5P, and this is actually one of the earliest identified cancer-promoting microRNAs, or otherwise known as oncomeres. And it has been shown that in several oncology cases, this particular microRNA in fact activates the PA3 AKT signaling pathway, and that also enhances oncogenic KRAS activation, leading to tumor initiation and progression. Now, the next two microRNAs are very interesting to us. This is miR155-5P and miR223-5P. These are immune cell enriched microRNAs. In particular, miR-155 is it's from a lymphoid origin enriched microRNA. An example would be T cells. And miR-223 is expressed primarily in myeloid cells, for example, macrophages and neutrophils. And what we're seeing here is that those two microRNAs, together with the cells where they're expressed, they're infiltrating these two particular cancers here. So in fact, these two microRNAs can be used as biomarkers to identify these two different cell types, ones that are from lymphoid origin and others that are from a myeloid origin. And lastly, we have the scramble stained tissues showing the specificity of this new assay. And of course, showing no signal whatsoever. Next, this is an example of microRNA detection coupled with IHC, looking at a particular T-cell specific marker, which is CD3. Now, as I mentioned before, miRNAscope assay is fully compatible with immunohistochemistry, and if a researcher has previously optimized RNAscope-ish conditions, they can be easily transferred to this assay. Now, this is a very important technique, having both the workflow for ISH and IHC, because as I mentioned previously, this enables the identification of specific cell types within a particular tissue of interest. Now here, miR-223-3P is detected in red, and CD3 marker is staining in green, as I mentioned, T-cell marker. Now this, as I mentioned, miR-223 is a myeloid-specific microRNA, whereas CD3 is a lymphoid marker. So as expected, we observe detection of these two targets are mutually exclusive, given that they're coming from two different lineages. Moving on to the next application is neuroscience. And microRNAs have actually been implicated to play roles from early stages in development all the way to adulthood. In this particular image, we're actually showing detection of a neural-specific microRNA, miR-124-3P, and this is in the mouse brain. Now, this particular microRNA is expressed in early development and continues to be expressed at very high levels throughout the animal adulthood. Now, miR-124 actually plays a crucial role in supporting neuronal differentiation, and one way in which it does this is by targeting a particular mRNA called PTBP. We're also showing here detection of miR-124 across various species. So we have the mouse brain on the left, the rat brain in the center, human brain to the right, and also not shown here, but we have also looked at other different species such as non-human primates, horse, and even rabbit tissues. In the mouse example, we're actually seeing a very strong staining of miR-124 in the hippocampal region, which is right in the center of that particular image. Also, in the rat brain, we're seeing high expression in the cerebellum. And in the human brain, this particular section can be seen exclusively expressed in the human tissue. Now, again, to demonstrate the specificity of this particular probe and overall the miRNAscope assay, we're showing a negative tissue known to lack expression of this particular microRNA, which is a mouse liver. And this, again, shows the specificity of the probe and the assay itself. Moving on to, again, neuroscience, but now capturing other microRNAs. So in the previous two slides, we have shown miR-124, and now here we're showing it in a higher magnification image, demonstrating the high level of enrichment among neuronal cells. To its right, it's LET7B5P, and this is a member of the well-established LET7 family of ultra-conserved microRNAs. So this particular microRNA is actually expressed across the mouse brain and many other tissues. And it has previously been implicated in central nervous pathology, tumor growth, and also immune response. miR-138-3P, it's right next to it. And this particular microRNA shows a very distinct expression pattern in the Purkinje cells within the cerebellum, as can be seen in the higher magnification image. Now, this particular microRNA plays a role in cellular differentiation and maintenance. And this also shows a perfect example of the spatial distribution morphological context that is necessary to really understand the biology behind microRNAs. Also, we have miR-128-3P, and this is a microRNA that was detected in cortical region of the brain. Previously, it has been shown to be implicated in control of neurogenesis and synaptogenesis, and also in neuronal migration and outgrowth. Again, as a control to show the specificity of the probes and the assay, we have a scramble stained tissue showing negligible amount of signal. Now, moving on to developmental biology, and this is perhaps one of our favorite images. This is in the showing the mouse E15.5 embryo sagittal section. At the top, we have the brain or the head of the animal, and on the bottom, it's the embryo's body. Now, this particular slide, again, highlights the assay's specificity compared to its negative control on the right, and the exquisite spatial distribution of miR-124, specifically in the central nervous system. Also, it shows the assay's ability to retain morphological context, which is really critical to really understand the function of this or any other microRNA. Other microRNAs that are also involved in developmental biology, this particular microRNA, miR-145-5P. And this is a cartilage-specific microRNA. Again, this slide shows E15.5 embryos, and three key areas we're highlighting here. First, digits on the far left. Second, the somites in the center, and the ear structure on the right. Now, miR-145P is actually being detected at different levels across these different tissue structures. Again, this is necessary to understand how microRNAs are regulating different biological signaling networks. And miR-145P is actually one of its main targets is fecal transferase, or FUT1. And this particular RNA has been implicated in osteoarthritis, which is actually characterized by cartilage degradation and chondrocyte injury. We also show here on the bottom scramble stained tissues showing no signal for this particular tissues. Our next example is a microRNA in development. It's miR-182-5P, and it's being detected in the mouse eye, specifically in the retina. Now, this particular microRNA is it's originally from literature expected to be expressed in the outer nuclear layer of the retina, and this is the place where the cell bodies of the photoreceptors are housed, for example, the cones and the rods. Now here, miR-182 is shown in the outer nuclear layer with very little to no expression in the inner nuclear layer or the ganglion cell layer, which are the layers just below the red chromogen. miR-182 has actually been shown to be necessary to maintain adult cone photoreceptor and visual function. And in fact, in mouse studies, deletion of this particular microRNA results in retinal dysfunction. Next is the detection of the enriched vascular smooth muscle cell microRNA, miR-145-5P. Now, miR-145 is actually a regulator of VSMCs, and it does so by regulating the proliferation and migration by targeting two key signaling pathways, SMAD and the TGF-beta signaling cascades. Now, on this slide, we have actually captured three different tissues to show the specificity of this particular probe. We have the mouse lung on the left, the intestine in the center, and the kidney on the far right. As can be clearly observed, there's this particular microRNA is quite abundant, but it's also very specific to the smooth muscle cells found in these different mouse tissues. Next, we're going to switch gears and we're going to talk about a different class of small non-coding regulatory RNAs, and these are the synthetic type, and how the miRNAscope assay can address this particular application. These are the small interfering RNAs, or siRNAs, and as mentioned, they're synthetically designed, and they can target a particular RNA of interest. And in many cases, those targets include transcripts whose aberrant expression can lead to a particular disease state. Now, it's been envisioned that in the next few years, many siRNA therapeutics will be entering the clinic to treat a number of diseases. So what are some of the key applications where siRNAs are being utilized? First is in oncology, and siRNA therapeutics are mainly focusing on targeting oncogenes in a number of malignancies, and in doing so, in modulating transcriptional landscape in an attempt to control tumor growth. Next is neuroscience, and siRNAs are being evaluated as promising tools for acute brain injury treatment, as well as other neurodegenerative diseases. And finally, in immunology field, siRNAs can actually target genes that are involved in autoimmune disorders, and in this way, hopefully slow down disease progression. Now, miRNAscope is actually a perfect tool to detect this particular siRNAs and to evaluate numerous parameters associated with siRNA delivery. Within our assay services team, we've actually looked into different siRNA chemistries or modifications and how these modifications impact biodistribution, and also looking into dosage studies, as well as stability of the siRNAs in longitudinal studies. This is an example of a recent study that was completed through our assay services team. And in this particular example, we have an siRNA, which we're going to call for the purposes of today, siRNA1. And a particular cohort of animals was delivered this siRNA. A different animal cohort to the left was administered saline, or if otherwise, our negative control. And the far right tissue is showing an animal cohort, which was delivered the siRNA number 2 molecule. Now, this slide captures the specificity of the miRNAscope assay, as only siRNA1 is being detected with the siRNA number 1 probe, while there's no signal observed in either the saline or in the completely independent siRNA number 2 molecule delivered to a different cohort. Now, again, this slide shows the type of actionable data that can be obtained with the miRNAscope assay. This data can drive new hypotheses or questions that are related to chemistry of the siRNA, dosage, how does that impact stability, the biodistribution of the siRNAs, and of course, the impact on the target of interest. Moving on to the next synthetic oligo, where miRNAscope has made an impact, are antisense oligonucleotides, or ASOs. Now, these synthetic oligos are typically single-stranded in nature, and they're actually designed to target genes of interest. A research area where ASOs are continued to be evaluated is oncology. And in oncology specifically, they're being synthetically designed to target and block oncogene expression. In neuroscience, they have emerged as important therapeutic strategies. Now, ASO-based therapies are now used to target genes that were actually considered at one point undruggable. And many of these ASOs are actually moving through the clinical trial pipeline. And in some cases, some ASOs have actually already gained FDA approval for the treatment of neuromuscular disorders. Finally, in terms of immunology, synthetic oligonucleotides can target key immune regulators in a number of diseases, including autoimmune disorders. To show you an example where a miRNAscope assay specifically detected siRNA target of interest, we're showing here two different tissues, one mouse cardiac muscle and the one on the right, mouse quad muscle. Now, different animals were administered either a vehicle or saline treated, or they were administered the ASO of interest here. And this slide shows the specificity of the miRNAscope assay detecting the ASO in the treated only animals, whereas vehicle shows negligible amount of signal. Finally, our last application is detection of piRNAs. Now, these are by far considered the largest class of small non-coding RNA molecules. And as mentioned previously, they range between 26 to 36 nucleotides in length than traditional microRNAs. Now, piRNAs are involved in two key research areas, which include epigenetics and oncology. In terms of epigenetics, piRNAs and their associated effector Pwi proteins, they're called the guardians of the germline, as they maintain genome integrity and fertility, primarily by preventing DNA elements from moving through different genomic regions. In terms of oncology, a number of piRNAs and their associated proteins have also been shown to be abnormally expressed in numerous cancer types. However, their function and mechanism is still very largely unknown. This is an example showing detection of 3 germline-specific piRNAs, piRNA1, 3, 13, 11, 21, which are the first 3 images in the mouse testes are shown here. And as I mentioned, piRNAs play a role in transposon surveillance mechanisms. Now, based on literature, these 3 piRNAs have actually been isolated and also validated as piwi protein interacting RNAs using a number of different approaches. Now, piRNA detection appears as very strong perinuclear puntate dots, and specifically in the spermatogonia, which is undifferentiated male germline cells that eventually give rise to mature spermatozoa. Now, this staining pattern is actually similar to that published in other literature reports. As a negative control, as we have continued showing, we have a scramble probe where there's little to no signal that can be observed in this particular mouse tissue. Moreover, we actually looked into other somatic tissues. As I mentioned, these piRNAs are germline specific, so we wanted to determine the specificity of these probes. We're showing here the mouse liver, a somatic tissue, and we have negligible levels of PW detection as expected. Again, showing the specificity and the sensitivity of our miRNAscope assay. And with that, I'll let Helly continue.
Thank you, Miguel, for the excellent application demonstration. In the next 2 slides, I'll talk about our recommended assay controls and scoring guidelines for the miRNAscope assay. The miRNAscope assay to detect smaller RNA will offer universal positive and negative control probes that will work for any species or samples. For the positive control, we recommend the small nuclear U6 probe, which is known to be expressed in most tissue samples. For example, on the top row here, shows strong nuclear U6 signal observed in a variety of tissues, including human and mouse tissues, and HeLa and 3T3 cell pellets. On the other hand, a universal scramble probe is recommended as the negative control probe to demonstrate assay specificity and to be used for data interpretation against your target. Next, we'll focus on the miRNAscope scoring recommendations. The miRNAscope assay data could be analyzed with either SEMI-QUANTIFICATION or single-cell analysis with a software analysis tool such as Halo or other open-source software analysis tools. The SEMI-QUANT scoring criteria shown here is specific for the miRNAscope assay, which provides guidelines for target expression levels as counted by dots per cell. If your target is highly expressed, such as with 20 dots or more per cell, then we recommend a score of 3, whereas if your target is moderately expressed with 11 to 20 dots per cell, a score of 2 is assigned. For lower target signal detected, such as 2 to 10 dots per cell, a score of 1 is assigned. Lastly, a score of 0 is assigned if there are no staining or less than 1 dot per cell observed. A note that we want to mention is that due to the relatively small size of the microRNAs or ASOs or siRNA targets, the puntate dots seen under the microscope may represent multiple copies. As a result, we recommend our users to interpret the data by relative quantification, meaning that they should always use the negative control as the baseline for measuring the change in their target probe expression. Next, I'll go over the miRNAscope products that are available. As mentioned previously, the miRNAscope assay is offered in both the manual and Leica formats. Each assay with universal control probes that are species independent, but are platform specific, so be sure to request the correct catalog number for the assay format that you prefer. For the manual assay, we have the HD reagent kit that includes all the pretreatment reagents along with the detection reagents needed for the assay. In addition, we also offer 2 introduction packs for the most popular species samples, such as human and mouse, for your convenience, where the packs include the pretreatment reagents, detection reagents, control cell pellets, mounting medium, and a hydrophobic barrier pen that are necessary to complete the entire assay. On the other hand, for Leica users, we have the LS reagent kit, where it includes the pretreatment reagents, wash buffer, and detection reagents. The miRNAscope target probes are designed in the S1 channels that are specific for this assay and cannot be interchangeably used with the other assays such as RNAscope or BaseScope. We're very delighted to also share with you that we have a rich list of miRNAscope probes pre-designed as catalog probes, so you can request them immediately. Feel free to reach out to your local account manager or our support team for the list of probes available. For custom design requests, we request you to submit your target through the ACD's new probe request web document on our webpage that's listed on this slide. In addition to products, we also offer in-house assay services for those of you who would like to send in your samples. We understand the challenges that many of us are facing during the pandemic. However, research must go on, and that's why our services team are here to assist you with moving your research forward. Our pharma assay services are fully operational and could help you advance your research in the detection of microRNAs, ASOs, or siRNAs. If you'd like to send in your samples and prefer the assays to be validated by ACD miRNAscope assay experts. Our services team ensures all due care is taken with the samples and that the assay development and analysis are conducted with highest efficiency with minimum time. Our pharma assay services has recently received awards from the Pharma Tech Outlook for being one of the top 10 CROs of 2019 and 2020 consecutively. In addition to pharma assay services, we also offer academic assay services for those of you who are working in the institution. The benefits of having ACD validate your target of interest in your samples include saving resources with your pilot study before setting up for a larger scale in your own lab, and receiving your results in a quick turnaround for your grant application or paper submission. For further information, please contact our support team with the email address listed here. To summarize today's topic, ACD's new miRNAscope assay is a robust RNA in situ hybridization assay, enabling the detection of smaller RNAs in tissue with spatial and morphological context at single-cell resolution. Key applications of ACD's new assay include ASOs, microRNAs, siRNAs, or other short RNA sequences that are 17 to 50 nucleotides. The new assay is a single-plex chromogenic fast read assay that is compatible with manual platform or the fully automated Leica platform. The miRNAscope assay is also compatible with IHC or IF, staining for the visualization of RNA and protein simultaneously on your tissue. Again, in-house assay services are also available for this assay if you would like our expert scientists to perform the assays for you with fast results. With that, I would like to thank everyone for attending today's webinar. For further details of ACD's newest miRNAscope assay, please feel free to visit our website or contact your local account manager to learn more. You could also contact our technical support team for any questions you may have regarding the new assay. The support contact information is listed here. So now we'll address some of your questions live.
Thank you, Helly and Miguel, for that excellent presentation. And now we will move on to the panel discussion portion of the event. I'd like to welcome back our speaker, Helly. And now we have excellent questions in our inbox. So let's start with our first question. How do you determine the specificity of miRNAscope target probes?
Hi, Amrita. Can you hear me okay?
Yeah, it's fine, Helly.
Thank you. So excellent question. So the miRNAscope assay leverages our core RNAscope probe design strategies and also the background suppression system to provide high specificity. And additionally, the target probe is screened through the whole transcriptome of the sample species to avoid any cross-hybridization. And also control samples are always recommended to be used to further evaluate the specificity of the probe.
Oh, thank you, Helly. Okay, second question. Do we have a list of target probes available to purchase? And also, could we design probes for custom sequences?
Yes, we do have a list of miRNAscope probes that have been pre-designed with a lot of popular microRNA targets for the research, and they're available for purchase immediately. You may also submit custom sequences for probe design feasibility through our new probe request form on the acdbio.com website.
That's great. What would be the lead time for the design?
The lead time for the design will take about 3 to 5 business days.
Okay, thank you, Helly. Next question. Can the miRNAscope assay detect RNAs that are greater than 50 nucleotides?
So the miRNAscope assay is specifically designed to detect smaller RNAs that are 17 to 50 nucleotides. For any RNAs that are greater than 50 nucleotides, we would recommend the BaseScope assay, where it supports sequences that are 50 to 300 nucleotides. And for any RNAs that are greater than 300 nucleotides, the RNAscope assay would be recommended.
Okay, excellent. And the two platforms has to be used in serial sections, right?
Correct.
Okay, great. Is this assay compatible with IHC or IF, like the protein detection?
Yes, the miRNAscope assay, similar to RNAscope and BaseScope, is compatible with IHC or IF for RNA and protein detection simultaneously in a single sample tissue.
Great. And ACD does have manual for that.
Yes, we have tech notes available for the detection of RNA and protein.
Okay, great. And is this assay workflow different from RNAscope?
The miRNAscope assay protocol is very similar to the RNAscope singleplex assay protocol. Specifically, if you're familiar with the manual RNAscope protocol for single detection, the manual miRNAscope protocol is very similar, but with an extra fixation step to ensure optimal detection of these smaller RNAs. On the other hand, the miRNAscope Leica protocol is very similar to the RNAscope Leica singleplex assay, which can be completed with an overnight run.
Okay, great. Good to know that. Okay, if there are any, we are approaching the end of this event. So if you have any more questions which are not answered, we will get back to you via email. So now we are going to wrap things up here today. I want to thank everyone for attending and our speakers for their excellent presentations. If you have missed part of the presentation or would like to rewatch, a recording of this presentation will be landing in your inbox very soon. Thank you everyone for joining us today.